Okay, just to go over what I just said to everyone during the Google Meet. Again, talking about the structure of DNA. DNA is made up of smaller units called nucleotides. A nucleotide has, in the middle of it, has a sugar. And so for DNA, it's deoxyribose is the sugar that's in the middle of the DNA. Also on one side of it, attached to the first carbon of the sugar, is a nitrogenous base. And that nitrogenous base will change depending on which nucleotide you're looking at. One of them would have adenine, one would have thymine, one would have cytosine, and the other would have guanine. Okay, and then on the other side of this deoxyribose sugar is a phosphate attached. So at the carbon 1 is the nitrogenous base, at carbon 5 is the phosphate. And then in the middle here at carbon 3 is where this ribose, or this deoxyribose rather, is going to attach to another phosphate. And so we're going to look at that. That's what 3' and 5' means. Along the backbone of the DNA, you've got some deoxyribose attached to phosphate. And the order in which they're sitting is based on whether or not the phosphate's on one side and the ribose is on the other. That would be a 5' to 3', or if it's turned around on the other side of the DNA and it's 3' to 5'. So we're going to look at that. The way that DNA is read by the enzyme that puts it together during replication is it's read up 3' to 5', and then it's pulled together the opposite direction, and I'll go through that. So 5' to 3' is how it's replicated. And remember that replication happens during mitosis and meiosis. So during that S phase, that synthesis phase of mitosis and meiosis, when the cell's getting ready to divide. Okay, so let me go to what that means. So for example, if we had a piece of DNA that was C, let's see, let me check my colors here. C, G, A, G, T, A. Let's say that that is the code for this little piece of DNA, for this tiny little part of the gene, okay? Usually genes are many of these long, okay? Like maybe hundreds of these long. But we're just going to look at a tiny little part. So it would look like this, okay? So this is cytosine. It's attached to its sugar and phosphate here. This is one nucleotide that has cytosine on it. This one has guanine. This one has adenine. This one has guanine again. This one has thymine. This one has adenine again. And so it's in this order, okay, that I just wrote down. And so then what happens is that there's a complementary strand on the other side of DNA. Remember, DNA is like a ladder with these rungs in the middle. Well, these rungs are just made up of the nitrogenous bases that are attached together. So these would have complementary strands, okay? And they're attached to each other with hydrogen bonding going on in the middle. And I went over hydrogen bonding in our meet. Hydrogen bonding just means that one part of these nitrogenous bases are slightly more positive and another part is slightly more negative. Usually the more negative part are like the oxygen and the nitrogen. They pull on the electrons more than the hydrogen does. So hydrogen are slightly more positive because they don't have enough protons in their nucleus to pull strongly on the electrons. Now oxygen and nitrogen do, so they pull on the electrons more. And we talked about this in class when we went through covalent and ionic and hydrogen bonding. So because of the slightly more positive hydrogen and slightly more negative oxygen and nitrogen, these are attracted to each other. And this attraction is not a strong bond. It's not as strong as the covalent bonds that make up the backbone. So where the ribose or the deoxyribose is attached to the phosphate, that's the covalent bond, and it's very strong. They're sharing electrons there. Here they're not sharing electrons. Here they're only being attracted to each other because they're opposite charge, slightly opposite charge. Okay, so that's a very easily breakable bond, and that's what happens during DNA replication. This gets broken up by an enzyme and pulled apart, and other enzymes will hold it apart, other enzymes will get it ready to go, but then the enzyme DNA polymerase is going to come along and attach the different parts to it. And so what happens is that it starts at the, at the 3' end. It's going to read it from 3' to 5'. But the parts that it's attaching start at the 5' end. So for example, we're going to attach a thymine to this adenine, and it gets attached like this. And so see how this starts with a 5' and goes to 3', but it's being read from 3' to 5'. All right, so they would attach that one first, and then they would attach, because that one is thymine, we're going to attach an adenine to it, like that. And then these, so these are getting attached here with some hydrogen bonding. And there's another enzyme that comes along and makes sure that these get bonded together, and they glue it all together. Okay, the next one, because that's got a guanine on it, we would attach a cytosine to that. Okay, and so again, these are getting attached here. The hydrogen bonding is going to keep the complementary pairs together. So that happens in that direction from 5' to 3'. It's being read, though, from 3' to 5'. And then on the other side, the same thing's going on, but it's being read again from 3' to 5' and being attached 5'. So let's just start with this one. Let's look at, because it's kind of random how it works, but there are different enzymes that figure out where to start the point of attachment, and I'm not going to go to the in-depth to explain that to you right now. But anyway, so we saw that this one went, and I accidentally blew it out, but this one went from this direction here. This one's going to go this direction, and so we would take this one and then this one, and see, I'm adding them from 5' to 3', but it's being read from 3' to 5'. Okay, so that's what we went through also. So so during DNA replication, this double strand gets broken up, and complementary bases get paired up with it in a 5' to 3'. I'm sorry, in a 3' to 5'. It's read that way, but it gets added on 5' to 3' on either side. So this one's going this way, and this one's going that way. Now, what does that mean? We talked about how it's semi-conservative. And so the word semi means partial or half. Conservative is kept. So these two parts are the old DNA. When it gets opened up, they're kept. Okay? And then new DNA, the new half is added, and these are going to be the new coming in, the new nucleotides being added to it. So when it gets completely done, you're going to have two different DNA strands. This one will have the complementary bases added to it on this side, and this one will have complementary bases added to it on that side. And so you'll have two completely ready to go DNA strands. And it's a lot more complex than that, because like I said, there are a lot of enzymes involved that we're not going to talk about. But I did want you to understand a little bit about how it's bonded together, how it's semi-conservative, and the different parts of the nucleotide. Again, understanding that you've got deoxyribose in the middle with a nitrogenous base attached to this first carbon. You've got phosphate attached to this fifth carbon, and the bonding that happens along the backbone of the DNA, or along the outside of the ladder, if you will, right? This backbone. Okay, this is composed of the deoxyribose and phosphate, okay, and so that's what these two are. Okay, these two parts of the molecule are along the back, and then in the center are the different bases, okay, that I've color-coded for us today. All right, I'm going to post this, so if you did not get to come to the Google Meet, hopefully I went over some things that will help you. If you have more questions, you can use a link that I sent you to sign up for some individual help time. I have office hours on Mondays and Thursdays from 1 to 3. I'm just sitting in front of my computer waiting for somebody to tell me that they need help. And so make sure that if you need help that you come in, that you contact me. All right.